This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 62, for December 11th, 2009. Hey everyone, it's Vincent Racaniello on a frigid day in New York City. And with me is Dick Despommier.
Hey, Vince.
How's it going?
It's going very well.
Pretty cold this morning.
It's pretty cold.
But I bet it's colder up in western Massachusetts where Alan Dove is.
You know how cold it is?
No.
It's so cold that all the politicians had their hands in their own pockets.
Oh.
Hey, Alan, how you doing?
Doing all right. How about you?
We're okay.
Yeah, it is indeed chilly up here.
Yeah, I would guess you're 10 degrees. So this morning at 7 or so, it was 5 degrees, -5 centigrade.
Really?
Yeah.
Wow.
Do you know what it was up there, Alan?
It was 19 Fahrenheit. So it was, I've got a converting calculator here somewhere.
Give us an approximation.
Converting calculator. You go to Google and you type 19 Fahrenheit in centigrade, it'll tell you.
Right.
Oh, okay. It was -7.2 in Celsius.
Wow.
Yeah.
Cool. Well,
Cold.
glad to have you back, Alan. Hope you've not missed viruses too much.
Well, no. I had some around the house for a couple of days, and
You haven't written about any lately, have you?
no, the blog has been a little inactive, and I was busy with work and had a couple of other issues come up. I had a death in the family, and then
Right. Sorry for your trouble, by the way.
thank you, and then my daughter brought home some kind of respiratory virus from daycare.
Well, you're in the right show.
Yeah.
All right. Well, the holidays are coming, so flu season should be beginning strongly.
Yeah.
Again, we've peaked, and now I bet it goes up as people travel for the holidays.
Yeah, the good news is that now anybody who wants it can get the H1N1 flu virus.
Vaccine?
Yeah, it's available to everybody now.
Have you gotten it, Dick?
I have not, because it hasn't been available where I've been.
Yeah, here at the medical center, we have not been told it's available to us yet.
That's right.
But did you see the CDC just released some new numbers on the cases, new estimates? We'll put a link to this. Up until from April to November, total infections, 47 million.
Right.
What's the population of the U.S.?
310 million.
So that's a good fraction, right?
Yep.
Of the U.S.
It is.
47 million. They have a nice distribution, 0 to 17 years, 16 million. Hardest hit, 18 to 64 years, 27 million. Maybe there are just more of them.
People in my age group don't get it at all, it looks like.
4 million people 65 and older. You just gave away your age, Dick. I thought you were 45.
I make no secrets about this. I am what I am.
And what would that be?
And that's all that you am.
69.5
Cool.
and counting.
I'm honored to be...
What is that in Celsius?
What is that in dog years?
Hospitalizations, 213,000 and almost 10,000 deaths.
Yeah.
This is just the U.S.
Right.
So those are the...
And that's just H1N1.
Right.
It's exactly right, because as you know, there isn't much of anything else in terms of flu.
Thank goodness.
Looked at the numbers this morning. There's nothing H3N2 or seasonal H1N1. So this seems to have squeezed those out.
Mm-hmm.
Well, but
yes.
now we're just getting into what would normally be the flu season wrapping up. So it'll be interesting to see if we do get a wave of seasonal flu.
Yes.
No, you're absolutely right. If you look at the charts on CDC, we've had a peak which began at the end of August and now has gone down. But that's not the real flu season. That's not coming till now December.
Right.
So that's a good point. We'll see what happens. I'm sure we're going to have more influenza.
Oh, maybe there'll be some herd immunity here.
Maybe something else. Some people think that we've immunized or infected enough people that we are really restricting the spread severely, which may be.
Yeah.
And it could be that now you have to have mixtures of people in new ways,
Yeah.
like what happens around the holidays, right? So
Right.
we'll see what happens. TWiV will keep you posted, right?
Indeed.
We'll keep a finger on it,
You bet.
or a respiratory tract, as we say.
But we'll wash the finger first.
Oh, yes.
There's a list on the board here. We're having a holiday party next week down the hall, not here, Dick. And everyone has to bring something. So I was going to bring hand sanitizer. But then they said you have to cook something that's from the country of your background. So
Oh, Vince.
I'm not from hand sanitizer.
So, I think with a name like Racaniello, you have a wide open slate.
Yeah, I'll bring some pasta. Yeah.
Sausage and peppers.
Well, I'll keep the meat out so everybody can eat it,
Oh.
Anyway, we have 3 stories today.
Okay.
HIV, West Nile, and flu.
Right.
Yeah.
So the first one is very interesting. We talked about the HIV vaccine failure some time ago. This was the PAVE trial. That was the name of it, or STEP. I don't know. STEP.
This was the STEP trial.
Not PAVE. PAVE is different, right?
Right.
So this was a STEP trial that was run by Merck, I believe, right? And it was called off. Let me get the reference up here. Where did it go? Do I have a reference?
They stopped it in September 2007.
I do have the reference here. September 2007, H1N1. Oops, I didn't open the PDF. Let's do that. This is a PNAS paper. Here we go.
Yeah,
So
they did an intermediate analysis and called it off.
I called it off because the people getting, some of the people getting the vaccine were showing higher rates of HIV infection. So they called it off. And this was an adenovirus vectored vaccine. So they had pieces of the HIV genome with relevant epitopes in adenovirus vectors, adenovirus 5 and adenovirus 11. And what they found was, the trial was halted because individuals that were seropositive for Ad5, this is before the trial began, because adenoviruses are ubiquitous viruses, so many people are infected. Those people showed increased rates of infection when they got the HIV vaccine, increased rates of HIV infection, so that wasn't good. Now, this paper is called Adenovirus Vector Vaccination Induces Expansion of Memory CD4 T cells with a mucosal homing phenotype that are susceptible to HIV-1. Very interesting hypothesis. So this group proposed originally that the reason these guys got more HIV after being immunized was that they were adenovirus-specific CD4 cells that were homing to the mucosa, and then they would be a nice target for HIV because that's where HIV replicates, right? So that was their hypothesis. So they did a study here basically where they took 20 individuals and took lymphocytes from them. They measured their adenovirus antibodies. They did cytokine profiles. They did stimulation of T cells. Basically what they find is that what happens is that these people that already had antibodies against adenovirus, when you boost them with the adenovirus vector, you get a memory response of CD4 cells. A memory response can be either antibody or cellular, and this is a cellular response. And these CD4 cells have a receptor on them. It's called alpha-4 beta-7 integrin, which allows them to home to mucosal surfaces. It's called the homing receptor. So the idea is that when you gave these individuals the adenovirus HIV vaccine, they already had pre-existing immunity against Ad5. They had preferential expansion of T cells, CD4 T cells, which are susceptible to HIV. They go to the mucosa,
which is close to where the virus is being delivered.
Exactly. Yep. And then you get more virus targets for HIV-1 and higher susceptibility to HIV acquisition.
Makes sense.
And they have a lot of data, which we won't go into. It's a bit complex, which really supports this. I mean, they did it with people. They took cells out of them and challenged them and showed that basically you have a memory response, a CD4 memory response, and they home to the mucosa. So why do they do that? Because adenoviruses infect your mucosal layers, your respiratory mucosa, your gut mucosa, and then your body remembers that. So when you get challenged again, it makes cells that go to those areas. Brilliant, isn't it?
Very.
It's because that's where it expects the virus to be. And in fact, the people who weren't adenovirus seropositive at the beginning of the trial, they didn't have a problem because even though they got immunized with adenovirus, they were immunized in the arm.
Right.
So they didn't make any cells that were homing to mucosal layers. Isn't that neat?
Yeah.
It's very cool. Anyway, I thought that was a great hypothesis. I was not aware of this. This paper just came out at the end of November. I wasn't aware of this hypothesis, but I thought it was very cool.
So the designers of the vaccine, to begin with, picking a vector should have perhaps considered this as an option of unintended consequences and opted for another vector that didn't home in on the very site that HIV-1 virus might actually encounter during an infection.
Yeah, well,
I mean,
I bet there were some people who said this was a bad idea.
yeah.
Yeah, well, in fact, I've been kind of skeptical of adenovirus vectored vaccines for as long as probably they've been around.
Yeah.
I'll take one against rotavirus.
An adenovectored vaccine?
Yeah, because the cells that are going to react against the rotavirus would be right there to intercept it. But this other virus infects the very cells that you would expect to be your immune system, right?
Right, but quite apart from this, I mean, you've got the issue that most of us, or a large percentage of us, have already been exposed to bunches of different adenoviruses.
Sure.
And the ones that are being used as vaccine vectors are often ones that are already commonly distributed in the population. So you're designing a vaccine with a vector that people already have immunity to.
Right.
And then you're putting it in and you're expecting it to work, which is kind of a hurdle. And then you're expecting it to not have any complications, which it seems like it's always seemed like a real stretch to me.
Yeah, and the answer has always been, well, we have 51 serotypes. We can always switch to another serotype if people have immunity. But that always seemed to me to complicate matters more than necessary.
So, in other words, the vaccine itself may have actually been a good vaccine. But the delivery system was screwed up.
Here's the thing. Remember the TWiV vaccine, that trial for HIV? That seems to have worked. That was a pox virus vectored vaccine and a naked DNA vaccine, no adenovirus.
Nope.
So I went back and looked at that, and that's why they didn't have this issue. So the last sentence of this paper is, our findings suggest a cautious approach in the development of adenovirus virus vector vaccines for HIV. Like, not at all, maybe?
Yeah. I mean, on the other hand, switching sides briefly, I kind of understand why people do keep using adenoviruses for vaccines, because they're relatively easy to work with, and they're probably not going to kill people.
And because everybody's had it already, there's no harm in giving it again.
But Alan, they did kill someone, right?
Yeah, there is that.
Yeah, so
Arg.
the pox...
This is not the first time we've heard bad news about adenovirus vaccines.
The pox vaccines are very compelling, especially the canary pox, where there's no human disease at all by the virus to begin with. But I think those are promising.
Yeah.
Or growing them in plant cells in a vertical vaccine farm.
Tell me about that.
About that. Anyway, that's cool. I like that paper. HIV vaccine failure probably explained, and we'll see what happens with adenovirus trials.
So they could actually go back and use the same construct for the HIV vaccine in a different vector. And
put it in a pox virus.
And expect to see a completely different result.
Yeah, you know what, Dick, though? These groups tend to be really compartmentalized. They probably won't give up and switch to the other group's vector because they have an investment in adenovirus or vice versa. It's really unfortunate, right?
And in many cases, there are also intellectual property issues.
Yeah,
exactly.
You
just can't pick up a vector and use it. You have to pay someone. It's too bad. It shouldn't be limited that way.
You're right on that one.
Before we go on, I'd like to acknowledge the support of TWiV by Citrix, the makers of GoToMyPC. Holidays are here, parties, shopping, time with family and friends. But for some of us, that doesn't mean that business stops. My solution for traveling during the holidays, or if you need time off, and you have to stay connected with your work, is to use GoToMyPC. To access your home computer while visiting others. You're going to see your in-laws. You don't want to schlep things along with you like your laptop. You can access any file or program on your office computer using GoToMyPC. You don't need to haul your laptop with you or copy important documents to a flash drive. GoToMyPC gives peace of mind because I can access anything on my computer from anywhere. Our listeners can try GoToMyPC free for 30 days. That's a month of unlimited remote access for free. For this special offer, you must visit www.gotomypc.com/podcast. That's gotomypc.com/podcast. You'll get a free 30-day trial. And we thank Citrix for their support of teaching viruses to everyone. TWiV.
Yes, indeed.
Ditto.
Dick, here's a story for you. This was sent by Len, who
I've actually read this.
Len said Dick is going to like this one.
I love it. I've already read it, and it's a fantastic article.
So people who have been infected with West Nile may have persistent virus in their kidneys for years after infection.
That's quite amazing.
Let's get this article up here. It's from the Journal of Infectious Diseases.
Fairly reputable
from a group
journal.
in Houston, Galveston and Houston.
Mm-hmm.
So they say when West Nile reached Houston in 2002, they established a longitudinal study to follow people who were hospitalized after West Nile infection.
Mm-hmm.
And they have followed 112 patients, and they evaluate them every 6 months.
Right.
For clinical symptoms, and then they look for virus. And they say here, a year after infection, 60% of the patients still had problems, symptoms of some sort. In particular, encephalitis still.
Well, it does kill off brain cells.
It does. And they say resolution of symptoms plateaued 2 years after infection, and after 5 years, still 60% of the patients with encephalitis still had symptoms. So the ones who originally had encephalitis, 60% of them still had some kind of clinical symptoms.
Sure.
So these people, apparently there were some kidney problems. Let's see what the story was.
A couple of the patients died of kidney failure.
That's right. Deaths of 5 were attributed to chronic renal failure. So they said, let's look in the urine for West Nile virus. So they did some RT-PCR, and in fact they did find virus in the urine. Now, how do I get to the second page of this article? There's no thumbnail showing. Well, let's see here. View thumbnails. Where are they? Here we go.
The PDF just opens in my browser.
25 patients, urine from 25 patients, and 5 were positive for West Nile sequences. And look at the days after infection. 2,422, 6.6 years. Yep. So 3 of them were 6 years plus, 1 was 4 years plus, and 1 was 1.6 years.
That's amazing.
And they have various ages. They're all pretty old, 45, 70s, 60s.
That is the age group. That is the age group that's most susceptible to the pathological effects of West Nile.
Well, and they also all had hypertension.
Yeah, they all had hypertension.
So their kidneys were already under some stress.
So that's why. That's a consequence of the kidney disease, right? Yeah. Interesting.
Well, no, I think...
No, the other way. I think Alan meant it the other way.
Yeah, I think I meant it the other way. I believe...
They had a history of hypertension.
They had a history of hypertension.
Yes.
Yeah, okay.
So, you know, for every case that enters the hospital, there are about 200 inapparent infections in nature. Mm-hmm. So that tells you that there's a subgroup of people that are more susceptible to the pathological effects of West Nile.
Sure.
And so these may be those subsets that you're looking at now.
Right, and the patients that they're looking at are only ones who came to the hospital.
Right. So
highly selected. They've already selected the group.
That's right. That is exactly right.
Most affected by the disease, and they
right.
And all the ones who tested positive in this case not only had hypertension, history of hypertension, they also had encephalitis when they got the West Nile virus.
That's right.
So now, what would you like to see happen next?
Bigger study.
To include?
I'd like to go different geographic areas, all ages, not just people with encephalitis, but anyone who was infected.
How would you know that?
And had some kind of symptom and was diagnosed, I guess.
Oh, well, how about people who are antibody positive for West Nile but have never had any symptoms?
Yeah.
Yeah, that would be good.
I would love to see those people thrown in here as an infection control.
So, and it'd be interesting to see if any females show up, too, because all 5 of these patients also happen to be male.
Right. It is a good point. I want to read this sentence because this really addresses something we deal with on every TWiV. Additionally, all 5 of our positive individuals were older men, and we are unsure at this point whether this is a significant finding or simply related to chance.
Yes.
Right.
How many times do we hear a story of 2 people getting something and everyone thinks it's because of this or that, right?
Right.
Yeah.
These guys understand chance versus causation,
That's right.
Please remember this. We have a story later which is sort of related to this as well.
We're pointing out these correlations in this data set, but it's very important to recognize this could be purely random. It could just be you got 5 older men who had, they're older men, they're likely to have hypertension.
If they're African American, they're likely to have it at a higher rate as well. So I wondered if they identified that as a confounding feature.
Yeah, there might just be no. This might be entirely random that it happened to be these 5 patients.
Yeah, that's a good point. They don't mention that here. But
which is why you need a larger study.
you
Absolutely.
need the fact that there is virus after 4 and 6 years.
That's remarkable.
Is unquestioned.
Yes.
That is
Yes.
clear, and that's unique. And
that is the interesting part.
They say that's the first time that we've shown long-term persistence of West Nile.
Right.
And the public health impact of these findings is considerable.
I'll say.
Why do they say that, Dick?
Well, because these could be reservoirs for other infections.
Exactly. Yeah. You have these people shedding virus. So, in fact, they tried to find virus in the urine, infectious virus, but they couldn't do it.
Right. Just snippets of DNA.
Yeah, by PCR. So they think that the virus degrades very rapidly in the urine. So they're going to, but I'll bet they get it eventually.
Yeah.
But that's very interesting. So this brings us to a consideration of why this virus would be able to persist, right? What has to happen for a virus to persist?
It has to replicate.
It has to replicate under the radar
That's
of the
right.
immune system.
That's right.
It has to either be so low down that it's not detected, or it does something to actively block the immune response, right?
Now remember, we're an aberrant host here, basically.
Yeah. And the kidneys are a pretty bad place to hide out, actually.
That's true. And very few of infections in human beings result in a viremia high enough for a mosquito to actually take the virus up from the blood.
Mm-hmm.
And I don't know of any mosquitoes that actually drink urine.
Now, what else? The other thing that would be interesting.
Sorry about that.
The other thing that would be interesting, I just thought of this, is if they can get autopsy, maybe brain sections from the ones who died of kidney failure.
Good point.
It occurs to me if these folks are developing encephalitis, you've got probably virus in the brain, which is a very good place to hide out from the immune system.
That is true.
Yeah, that's a good point.
So it might not actually be hiding in the kidneys. It might just be we're seeing it in the kidneys.
Right.
And it's actually there may be a reservoir of it in the brain.
But my speculation is that this doesn't play any role in the natural history of the spread of West Nile.
You think so?
Yeah, because human beings in general, even little kids, although it's been speculated by some that little kids can act as a vehicle for mosquitoes transmitting it from person to person without having to amplify it in birds in places like Egypt or South Africa. But for the most part, adults can't achieve a level high enough to transmit that to an insect, and horses can't either. By the way, they don't achieve the viremias necessary to infect the mosquitoes.
Mosquito, yeah, because clearly it's not going to be transmitted by the urine.
No,
it's a very rare route in people, right? Except in healthcare settings, maybe.
Right.
Very rare.
Right.
Yeah, that's a good point. It's worth pointing out that in animal models, this virus has been shown to persist.
Yes.
In brain tissue.
That's
hamsters, they cite here. So now we know that that's relevant to people. So that's a good model to study this. This is very interesting to study, right?
I would say.
Could write a grant on this right away to figure out the mechanism.
Let's go for it.
I'm telling you, from TWiV, I get so many ideas from experiments, but unfortunately, you can't do them all.
Nope.
All right, that's a cool study.
But maybe our listeners can.
So, Len, thank you for that story.
Yes.
And Dick did like it. I could tell by the smile on his face.
He loved it.
Yeah. You going to ever revise your book, Dick?
Yeah. You mean the book on West Nile?
Yeah.
Yes.
And you can put all this stuff in it. You
Absolutely.
just go back to the old TWiVs and take out all the new stuff. And
Well, we'll do it something like that.
All right, the third story we have is another interesting one. This is now influenza. Adaptive strategies of the influenza virus polymerase for replication in humans. And this is a paper that just came out in Proceedings of the National Academy of Sciences. It's called Adaptive. I just read you the title. Here's the story. Now, we have to do a little background information here. Influenza viruses carry into the cell an RNA polymerase, which we talked about on RNA synthesis TWiV some time ago.
Yep.
Because the RNA is negative stranded, so the cell doesn't know what to do with that. So it brings in its own polymerase to copy it. The polymerase is made up of several proteins. There's a PA protein, a PB1, and a PB2. Okay, funny names, but P stands for polymerase. Now, in influenza viruses that you get from birds, there is a specific signature amino acid on the PB2 subunit at position 627. It's usually a glutamic acid in influenza viruses from birds. Okay? In most human strains, that's a lysine. That's 627. All right? And it's a very interesting site. This group has previously shown that So the avian influenza viruses don't grow well in human cells, in part because they have the glutamic acid at 627. If you change it to a lysine, they grow better.
No,
right?
Nature is so finely tuned.
It turns out that there's a protein in human cells which is binding the polymerase and inhibiting it if it has the glutamic acid, but not the lysine. They say there's a restriction protein in cells. They haven't identified it.
Huh.
Now, how about 2009 H1N1? What do you think it has at 627? It has the bird sequence. It has a glutamic acid. So how come it grows in people so well? That doesn't make sense because...
If that's correct.
I think that... they and others have looked at this 627 and looks like it's pretty important. But you're right. There could be other things impacting on this, right? So what they did in this paper was to say, is there something else in the PB2 of H1N1 that is making up for having a glutamic acid at 627? You got that? Is it clear? It's a little complicated.
Yep.
So they have a really nice assay. So you can take the genes encoding these P subunits, PA, PB1, PB2. You put them on a plasmid, you put them in a cell, and that makes polymerase, and then you can assay polymerase activity. It's cool.
Yeah.
So they can put the 3 plasmids in human cells or in bird cells or other species and see the effect of specific changes at 627. Okay? Okay so far? So we're going to assay these in cell culture.
Roger that.
So what they find is in human cells, if you have a glutamic acid at 627, you don't get much activity. And in a bird isolate, if you change that to a lysine, now you have a lot of polymerase activity. The 2009 H1N1 has pretty good activity, but not as good as if you changed it to a lysine at 627. So it could be better. So then they said there must be something else that's allowing the H1N1 PB2 to be a good polymerase in human cells. So they looked at the sequences and they compared all the isolates of 2009 H1N1, and they identified 2 amino acid changes that are conserved in all these isolates, a serine at 590 and an arginine at 591 in the same protein. They call this the SR polymorphism. And it turns out that that is why the 2009 H1N1 polymerase can work in human cells, even though it has a glutamic acid at 627, because it's got this other change, these other 2 amino acid changes. It turns out that all the H1N1 isolates have this other SR polymorphism, and that's why the polymerases work.
And if you put this polymorphism into an avian virus, then you get that polymerase will then work.
Exactly. Even though it has a glutamic acid at 627, it will work, yes.
So H1N1 has found a different solution to the same problem.
Exactly.
Safe crackers. They're all safe crackers.
Cool. Yes.
So I thought this was really cool. And it's just all... They actually also do infection studies. In the end, they put these changes into virus and show that everything makes sense in human versus bird cells. So if you have in an avian influenza virus, if you have a glutamic acid at 627, it doesn't grow well in human cells. If you change it to a lysine, it grows well. And these SR polymorphisms have similar effects. Very cool. So does this mean anything for the current virus? Well, this virus found a way to get around changing 627. I don't know why, though. Why didn't it just change 627? There must be some other reason that it couldn't just do that, right?
Maybe in one of its other, it's was in pigs, it was in birds through its natural history. Maybe in some other setting, it needed the 627.
Could be. It could be. And then when it got into people, it couldn't change it because maybe there was another protein interacting with it that was important. They say here at the end, despite the presence of the polymorphism, the 2009 polymerases are still restricted in human cells, so they're not as good as they could be if you change 627 to a lysine. Our findings raise the possibility that the 2009 H1N1 virus may further adapt to replication in humans by acquiring a lysine at 627. Continued surveillance for mutations at this position is warranted. So that's a good point.
Interesting. That is a very good point.
So it could grow even better if that changed, and it hasn't.
Mm-hmm.
And they say up to the point of this paper, which was November, none of the sequences have changed at 627 to lysine. I don't know if that's still true or not. I mean, it's interesting. Apparently, it's good enough to transmit very well. right? So it doesn't have to get any more efficient. It's satisfying its evolutionary requirements, right?
Right.
So
there may just not be enough pressure for the virus to improve.
right. And that's really, in the end, what we need is just be able to spread efficiently from host to host. We don't need to kill the host, right?
No.
We don't have to evolve to greater virulence. I'm setting up the next story. We just need to be able to transmit and reproduce.
Right.
I don't know of any example where a increase in virulence has been necessary for evolution, unless it helps transmission.
Right.
Which they could be connected.
Yeah, but Vince, that's there are so many effects that a virulent organism could have on the populations of hosts.
Sure.
That there could be very good reasons, evolutionarily speaking,
right,
for eliminating a competitor from a given biome. I mean, it's a very complicated thing.
You are right. I stand corrected. Let me put it this way. There's no need to evolve towards increased virulence unless it has some impact on transmission. Right. If it had zero impact on transmission, I don't think. But you're right. It could have other effects that make it more virulent.
Yeah. Sure. And that could be a short-term or long-term effect.
Absolutely. Mm-hmm. Absolutely. So they have a nice table here in the supplementary data, frequency of the SR polymorphism. So far they have 579 sequences here, and 100% have one of the two SR polymorphisms, the R, and 99.8. So one virus so far has a different base at that position, but in 627, all 100% glutamic acid. Interesting. All right, let's move into email, and this actually will get us into another story. And the first one is from Justin, and we did half of Justin's last week, and then we stopped because we were running out of time.
We didn't do it, Justin.
We didn't do it justice. He liked your joke about, what was it? Deja flu. Deja flu. Justin, second part of his question. Perhaps another thing that would be worth addressing is the whole issue of the one Ukraine strain being labeled as a low reactor. That one has spread all over the net like wildfire. How can a single change at 225 cause a vaccine failure? All right, so we need a little background on this. I don't know if you're familiar with the 225 story, Alan. Have you followed this?
I had not, and then I read up on your blog in preparation for the show.
All right, so a couple of weeks ago, there were some severe cases in the Ukraine and some deaths, and a mutation in the HA of the H1N1 viruses was found. And this mutation's been discovered in a couple of other cases before. It's a change in the hemagglutinin at position 225 from an aspartate to a glycine, D225G. The talk on the net has been that this is bad because it's going to make the virus more virulent, and then they think it also is causing vaccine failure. So let me explain the story here. So the HA, of course, binds its receptor on the cell sialic acid. Human strains prefer alpha-2,6 linked sialic acids. Avian strains prefer alpha-2,3 linked sialic acids. Now, this position has been looked at in the context of the 1918 influenza virus H1N1, and Peter Palese has done this work a couple of years ago. And he's shown that the 1918 virus likes to bind. Here's a nice chart here for you, Dick. It has a D at 225, and it likes to bind alpha-2,3 sialic acids. If you change the D to a G, it expands it to bind alpha-2,6 sialic acids. And then there's another change that you can make at a different position that can restrict it to alpha-2,3 again. So the idea would be that this change in some way impacts receptor recognition. And the idea is that the human respiratory tract has largely alpha-2,6 sialic acids, but there are alpha-2,3 down in the lower tract. So the idea would be that by changing D to G in the virus H1N1 currently circulating, you would allow it to bind alpha-2,3 linked sialic acids, go deeper in the tract, and be more virulent. Okay, so that's the idea. Now, Peter Palese's work was done with the 1918 strain. What about the 2009 H1N1 strain? So I found a very nice article. It's actually a letter to Nature. Oh, my thing closed with all my papers. Let me open it up again.
A technological meltdown.
This is a letter to Nature. It's called Flu Receptors in the Human Airway. So basically what they did, they took samples of human respiratory tract and stained them with different lectins specific for either alpha-2,3 or alpha-2,6. As I just told you, alpha-2,3 sialic acids are only in the lower in the alveoli in the lower tract. But you know what? Alpha-2,6 are throughout the tract. They're in upper and lower tract. They're not just in the upper tract. So I don't see the first question I have here is why would giving the H1N1 the ability to alpha-2,3 make any difference, because it can already grow in the lower tract where alpha-2,6 is. Got that so far?
Yeah.
Sure.
Right? So that, to me, that doesn't seem to improve its virulence.
Right.
And it turns out that this virus, which is actually, and here's another study. I'm sorry, I'm making a mess of this, but there's a second study. Here we go. See, all my papers have closed, so I'm lost without all my papers. Paper in Nature Biotechnology, receptor binding specificity of pandemic H1N1 virus determined by carbohydrate microarray. Ah, I remember this one now. This is a very cool paper. A carbohydrate microarray is where you take different carbohydrate linkages and put them on a nitrocellulose sheet, like 50 different ones. And they did that, and then they combined virus to it and see what it binds. It turns out, to make a long story short, that the 2009 H1N1 will bind both alpha-2,3 and alpha-2,6 with the D at 225. So you don't have to change to a G to change its receptor specificity. Okay, so the idea that this is making it more virulent, I think, isn't correct. But see, here's another thing, and this relates to the previous story. There were a number of people in the Ukraine who died of flu, and they got the virus with D225G from them. And then some people who didn't die had D at 225. And from that, everyone is saying, clearly it's causing increased virulence. And people are saying, well, it can't just be coincidence, but it can be. That's the killer of epidemiology, coincidence, right?
Yes.
It could be. You have to look at more people. And in fact, this mutation's been found in other people, and it doesn't kill them.
Right.
So everyone is all upset. There's all kinds of buzz on boards all over the net about this, but I don't think it's going anywhere.
So what we have here is a theoretical concern based on this mutation that, theoretically this virus might be able to infect further down in the lungs. But even that theory doesn't really wash.
It doesn't wash, right.
The receptor for either form is distributed widely throughout the lung. So, and H1N1 appears to get down deep into the lung just fine as it is with the original version.
Exactly.
And then the second problem is, so you've got this theory that was kind of shaky to begin with, and then you also have no data to support it.
Right. Exactly.
So what's your concern here? I mean, what's
strike three? As I recall.
Well, I think the concern is that people don't look at all the data. They just look
any
at
data.
They look at any of it, just the Ukraine isolation. They don't look at the biology here and the carbohydrate binding and all that. So, and then the second thing is his point is actually about vaccine failure. So, for some reason, one of these isolates from the Ukraine that had the change D225G was labeled in antigenic tests as a low reactor. And this is something that, if you search for D225G low reactor in Google, you will get all the hits with respect to this. The idea being that it doesn't react well with antibodies directed against the vaccine strain. So it may have undergone antigenic variation. So these people, even though they may have been immunized, and I'm not sure they were in the Ukraine, that didn't protect them because this virus had a change, okay?
Right.
So we've got one laboratory test on one isolate of virus from one patient.
Right. But here's the other thing, Alan. D225 is not in the known antigenic sites of influenza virus H1N1. All right.
Strike four.
It's not a site that, if changed, will change the antigenicity. I don't know why it's labeled a low reactor. There could be some other reason.
Yeah, it could also be an artifact. Yeah.
Could be an artifact. These serological tests are often very difficult. So I haven't seen the data, so I'd like to see it, but that's what I would guess. So I said all this to Peter Palese. I said, what do you think? Do you think it plays any role in increased virulence, D225G? He said, no, not at all. I said, do you think a change at D225 could lead to vaccine failure? And he said, this is bull. Now, he's the expert.
It's only half a word, but that's okay. I'll take it.
It's half a word. He's politically correct.
I'm sure he is.
Now, he's the expert.
Yep.
I'm going to leave it at that.
Yep. But there
Not
is
that we're, I mean, even apart from the appeal to authority, just on logical grounds,
of course.
The data don't wash.
You're
The
right.
theory doesn't hold up. There's just nothing.
No.
There's nothing to stand on.
No, here on TWiV, we can take care of these things, right?
We can, or we can at least express them and allow our readers and listeners to judge for themselves. But
I think that's a good point. We put the facts out and let people judge.
Yeah.
We don't have to depend on Peter Palazzi.
no, you don't.
Although it is certainly interesting to note that somebody who has spent his life studying these viruses and has worked with this particular one.
Yeah. No, we put all the facts out. We'll put the links and people can look at it and ask us questions if they don't get it.
Now
go ahead, Dick.
In a previous TWiV, I forget how far back we went, but with the H5N1 strain, there is a difference in binding in terms of the upper respiratory tract and the lower respiratory tract.
Right. So the H5 will only recognize alpha 2/3 linked,
which is lower,
which is only found in the lower tract.
Correct. So in that instance, then it explains the very difficult infection pattern for this virus. That is, most people exposed to it don't become infected because they don't inhale it deeply and they don't get enough virus to begin with. Whereas little kids who pick up a sick bird and pet it until it dies, those are the ones that are catching this mostly.
Because they inhale it deeper.
Correct. And their respiratory tracts are shorter. So this may bring that up to a higher level.
And then once it gets down there, it causes serious disease because it's deep.
Precisely.
But it doesn't transmit because it's hard to get virus out of the lower tract into the air again.
Well, that's right. So my question, as a non-virologist, are we talking about the same sugars in this case?
Alpha 2/3 and alpha 2/6.
Okay, so we have two different forms of influenza, one of which we can explain the virulence, and the other one we can't.
Yes. No, this one, the H1N1 of 2009, will recognize both alpha 2/3 and alpha 2/6.
Right.
There is a lovely picture in this carbohydrate microarray paper. Here is this graph of the binding to three different viruses. These two are on the top are 2009 H1N1, and this is a seasonal H1N1. These are alpha 2/3 linked sialic acids in green. These are alpha 2/6 linked. So the H1N1 of 2009 bind both. The seasonal only binds alpha 2/6. So there is a difference in specificity, which may explain part of the pathology differences, but we don't know.
We don't.
But there's alpha 2/6 down in the lower tract already, so I don't know why it would make any difference. So yes, your point is well taken, Dick. Okay, so I think that's a good analysis of that. Thank you, gentlemen.
Sure.
Now we have a few more emails. Peter writes, "Gentlemen, I just listened to TWiV 53 where you discussed whether or not to do a segment or even a show for the folks on No Agenda. No Agenda keeps coming back. I used to be listening to both John Dvorak and Courier, and all I can say is you've come to the right decision. Both the hosts are, I assume, quite well-mannered gentlemen when off the air, but on the show their claim to fame is to be talking opinions. The more outrageous the opinion, the more discussion it will spark, the better for the show. Their listenership is therefore what you would expect it to be, always willing to jump on the next outrageous idea. Actually, if you did enter the forum discussion, I think there's a good chance they would point out that you've done work for the NIH, and therefore you must be one of them. There goes your credibility. You can't reach everybody.
You rotten analytical scientist, you.
Yes.
That's right. That's right.
All you think about is the truth, God damn it.
A more interesting thing came up here in Germany last week and really stirred some discussion. The Spiegel, a large periodical here, reported that obviously the government had ordered 3 different types of flu vaccines. Pandemics, an egg-bred vaccine with adjuvants for everybody, but a tissue culture-bred, non-adjuvanted vaccine for the military, and yet another vaccine without adjuvants for state government offices, which includes the Paul Ehrlich Institute, the German Office for Infectious Diseases. Now, obviously, this was not a single group deciding to buy those 3 different vaccines, but the military and the executive offices made their decisions for their staff, but all of this stirred the discussion about 2 classes of vaccine qualities, the better unadjuvanted for the more important, and the lower quality stuff for the rest of us. Quite interesting is the fact that even some people in key positions in the health authorities and practitioners, including IIRC, the head of the National Organization of Pediatricians, came out and said that the adjuvanted vaccine was not proven to be safe for small children or pregnant women. The case for children actually sounded logical. Their immune system isn't yet in a steady state, and overboosting it with non-proven adjuvants might be dangerous, as it could lead to autoimmune reactions. Any ideas on that?
How about overboosting it by sending them to daycare?
That's right.
Children have the most overboosted and overattacked immune systems of anybody.
You bet.
So my take on this is that the adjuvant in Europe, MF59, has been used for a dozen years. It has very similar components to the AS3 adjuvant, which I believe is in pandemics. Squalene, some vitamin E derivative, tocopherol, and some detergent. So I agree that AS3 hasn't been tested in pregnant women or small children, but I don't think it's going to end up being much different in terms of safety profile.
Also, news bulletin, very, very few pharmaceuticals of any kind have been tested fully in small children or pregnant women.
Correct.
Yeah.
Even drugs that are routinely given to small children and pregnant women. In very few cases, if you go back to the records on these, you will find virtually none of them have actually been rigorously tested in those populations, because it's ridiculously hard to set up such a trial.
Exactly. I was going to ask you why.
IRBs don't want to approve any of those.
Yeah, so what you do is you wait until it's been used by the general population for a while, and there's no harm, and it's used in younger and younger populations and people with different immune statuses, and then you make the logical inference that it's probably okay in kids and pregnant women, and if it's something life-saving or potentially life-saving like a vaccine, then you'd say, okay, it's a go.
Yeah.
That's just the reality of the way you have to develop these things.
So what do you do if you're in one of those groups and you feel it hasn't been adequately tested? What should you do, Alan?
I would... Now, my own feeling about adjuvanted vaccine is it's probably the better of the two, because it's likely to stimulate a robust response. I would be fine in the case of these adjuvants that have been tested for more than a decade in Europe. I'd be fine with my daughter getting one of them.
I guess everyone has to make their own decision and balance what they think are the possible side effects of the adjuvant versus the possible side effects of influenza.
Yeah,
right?
And at least in this country, vaccine manufacturers have been, I think, ridiculously accommodating. When you go to the pediatrician's office now, they'll give your kid, just as a matter of course, they'll give them vaccine that doesn't have any thimerosal in it.
Right.
Even though that particular component has never really been proven to have any bad effects, there's fear.
Right.
And so in order to encourage people to get their kids vaccinated, the children's vaccines now lack this ingredient. So I'm sure that when the FDA does finally get around to approving adjuvanted vaccines in this country for flu, that there will be the manufacturers will make the unadjuvanted form available as well, and pediatricians will probably have that.
Yeah, I agree that if people are afraid of something, you should take it out, because that way you can get more people immunized, and I think that's important in the long run. He goes on to say the way those vaccines were certified was discussed at length. There's a class certification for the high-yield base strain, and only a small part of the certification has to be done with the actual vaccine produced with surface proteins. But I assume that's standard operating procedure for flu vaccines, because otherwise you couldn't possibly have one ready for the same year's flu season, right? And that's correct. You don't have to go through an entire clinical trial for every seasonal flu vaccine. And he concludes by saying, all in all, this is a pretty bad image we're giving here. I think that this whole story has lowered the willingness to be vaccinated considerably. Just goes to show how it's not only right to do the right thing, but also to communicate it correctly. Thanks for the show. And that's correct. There is an awful lot of controversy about this, worries about safety and testing, and it's unfortunate, and it continues to this day, right? Yeah. And I don't know. What we try to do on TWiV is to give you the facts and not the rumors. So that's what we're doing our part. Here we are from John Dickson. We answered this last week, but we got it wrong.
Did we?
Yeah, I asked my wife. She told me we blew it.
What did we blow?
So we'll have to answer it again incorrectly.
Uh-oh.
And now that Alan's here, he can do it too. John writes, I'm surprised virtually every day when discussing with my friends whether they will obtain the vaccine for novel H1N1 and, more importantly, whether they will have their children inoculated. Usually their concerns of safety are nonspecific and ungeneralized, and I go through some of the basic numbers with them, pointing out that although H1N1 may not be particularly more fatal as to rate of infection, that it will infect more people and the deaths will skew to the young, and this will be noticeable. Several days ago, I got considerable pushback on this topic from an individual who contended that he personally was awaiting U.S. trials to be completed before he would obtain his shot. He really did not believe that the FDA could appropriately approve the safety and efficacy of vaccines without U.S.-based trials. I told him this was rubbish. Since this individual has some science in his background, I questioned that assumption, told him that many clinical trials for U.S. drugs are done with non-U.S. based clinical trials, and suggested that with his beliefs he should probably never ever receive a regular flu shot and should probably throw out half of his pharmaceutics in general, since they may have depended on non-U.S. clinical trials. I stated to him that I was not aware of any reason to think that there is an immunologic difference between a resident of U.S. relative to a resident of Australia, Japan, India, or pick a country for the purposes of clinical testing of vaccines, and in particular flu vaccines. I also told him he could wait for any kind of clinical study he wanted, but the safety and efficacy studies necessary for the U.S. approval of the current H1N1 vaccines had been completed and were in the process of reporting and publications. Am I correct? All right, so we said that you could test anything anywhere, but it's not true. For pharmaceuticals, they have to be clinical trialed in the U.S. to be approved in the U.S. Is that your understanding also, Alan?
I believe so. I'm trying to think now. I'm trying to think of a counterexample. I'm trying to think of something that was approved in the U.S. without any what?
Flagyl.
Was approved in the U.S. without any U.S. clinical trials, really?
Yeah. Mm-hmm.
So a blood pressure medication has to be trialed in the population because, as you know, that varies in different ethnic groups.
Yeah, for those that don't know Flagyl, it's metronidazole.
It's an anti-parasitic drug.
Yeah, it was tried in Europe a lot for trichomoniasis.
Well, my wife, who works for a major pharmaceutical company, said for most pharmaceuticals you have to have a U.S. clinical trial. For most,
I would agree. Yeah.
Vaccines are an exception. They can be tested elsewhere because the assumption is that the response is going to be similar. Even though there are genetic differences in immune responses, I'm not aware of them being country-based.
A lot of drugs that are all available in Europe are available here under compassionate use, I guess, constraints. And with enough compassionate use, without any side effects, the drugs then would consider that a clinical trial in some cases.
I see.
Right, and I think as a practical matter, it's kind of done that way for drugs that have been in long use outside the U.S. You know, the real clinical trial occurred someplace else, and then the drug company will do a sort of clinical trial in the U.S. just to get the approval.
Right.
Yeah. Well, she also said that most big companies do international trials anyway from the start.
Right. Yeah, they'll have it done in 9 countries at once because they want to get the approval in as many countries as possible right away.
When you consider the ludicrousy of the requirement to have it done in the United States, considering where we all came from.
Yes. All clinical trials should be done in Africa.
That's right. Or only in Native Americans.
Right.
Yeah. So vaccines are, the H1N1s have all been tested, and those are going to be published in December, I understand. And the 30,000 people are involved in that trial, so it's quite extensive. And the FDA has seen the data, and they've approved the vaccine based on it.
Right.
All right. A couple more. Frank writes, "Hey, TWivers, I listen to and enjoy your podcast, which I find quite informative, even for a layman like myself. I searched on your website, but couldn't find any mention of Baxter releasing a mix of H3N2 and live H5N1. And he puts live in quotes. Excellent. Is that because you didn't know about it, or because it isn't the kind of subject you want to cover, or," he gave us a couple of links, and in fact, we did cover it back in TWiV 23. And if you search for Baxter, you won't pick it up, but if you search for, I forgot what I used. Anyway, TWiV 23, Frank, thank you. Mike writes, "Hello, I just found your podcast. I love it. I listened to the polio show. I have a question about how viruses exist outside of a host in the environment. You mentioned that poliovirus can last outside of the host for 6 months. I thought that viruses could last very long outside of a host. You can't get HIV from a doorknob." I think he means to say, I never thought that viruses could last very long.
Right.
What is it about some viruses that allow them to last for a long time outside a host? Does it have something to do with their shape or the content of the protein coat? Thanks. Mike is from Delaware.
Take offense.
Actually, you're not from Delaware. You're from
Maryland.
Maryland.
I'm from Maryland.
Yeah, sorry. Well, we used to think that an envelope virus was unstable and an icosahedral shell was very stable. And in general, the icosahedral shells are very stable. But now we know that some envelope viruses are very stable as well. So we can't make that kind of generalization. But, the envelopes are susceptible to bile salts, proteases, and so forth. But influenza, some strains of flu can pass through the gut unharmed and others can't. And so we really don't understand. I would still stick to the general idea that envelope viruses are more or less stable than icosahedral shells, but the specific determinants, I don't know. Any of you guys have some thoughts on this? Looked at a lot of data. I don't understand what controls it.
Yeah, it doesn't. Back in college, I learned that it was, the envelope viruses were less stable, and then
smallpox survives for a long time too.
It does.
Yeah.
So
and also survival outside the host isn't necessarily going to correlate with transmissibility. I mean, you look at measles virus.
Sure.
Yeah. Also depends on the conditions. Like flu will last on banknotes longer if there's mucus on the virus as opposed to just virus.
Yeah.
So it depends on a lot of things. So we can't be simplifying it for you, but when we get, I don't know any specific determinants of stability except a capsid that is stable versus an envelope that is not.
But I guess the answer to the general question is some viruses can last a very long time outside the host and other viruses can't.
Yeah.
And beyond that, it's speculation.
Well, I mean, flu will last up to a few weeks on a dollar bill with mucus, but polio will last up to a year in the environment.
Right.
And that may be a difference between an envelope and a icosahedral capsid. All right, Laura writes, this is interesting. H1N1 is said to have genes from pigs and birds. So when the vaccine is given, are you also getting DNA or genes for pigs and birds injected into your body? That is my only concern. Please explain. Thank you. No, you don't have any genes from pigs and birds. You have genes from influenza viruses that infect pigs and birds.
Exactly.
Not pig genes, not bird genes.
No.
So the vaccine has no genes from pigs or birds, or even people.
Exactly.
And
also,
it just has viral genes. Okay?
Right.
I thought we had it. I mean, I like to get this because we have to explain to people what's going on, right?
Now, let's keep this episode in mind, and when we do a TWiP on the survivability of schistosomes, we'll come back to this concept.
Okay. Well, you can remember, right? Because I won't.
I'll do my best.
Last one from Mike. Just wanted to send a note of appreciation for the wonderful episode a few weeks back with Lynn Enquist. I was on a brief break and had to catch up. I know you have an audience with a quite diverse background in virology, but as a graduate student, I very much appreciate hearing the unfiltered experiences from scientific pillars. It's very inspiring to hear these successful life stories. I know you get a lot of praise from non-scientists for the accessibility of most of your shows, but I wanted to express my appreciation for this particular episode, which may have been a bit jargony for some of your listeners, but was a real treat in a podcast I already look forward to each week. He writes, several episodes back, I realized we share multiple alma maters. I'm attending graduate school at Mount Sinai in Dr. Palasi's department, although not in his lab, but I also went to high school at Northern Highlands in Allendale, New Jersey. It was quite the shock to hear you call out Northern Highlands, knowing that you had also studied in the same department where I spend most weeks listening to your podcast as I run my experiments. So I went to Northern Highlands also. I completed my undergrad at Dartmouth rather than Cornell, though. That coincidence simply would have been too spooky. Coincidence. Thanks, Mike. And that will do it for email. We're almost caught up.
We are.
Let's do a couple of picks before we wrap it up.
Sure.
You got a pick, Dick?
Well, there is a D.A. Henderson book that just came out on the history of the eradication of smallpox, which I think is probably noteworthy. Although I can't confess to have read it yet, it certainly is. It would give a very unusual and very insider look at how a noxious disease such as smallpox was handled.
Yep.
And all the machinations necessary to get it out of here.
Okay. I've actually bought it, but I haven't read it yet.
Uh-huh.
But we'll put it as a pick.
Excellent.
Because that's an interesting story, and he's the guy who engineered it, the eradication, right?
Yeah.
That could be good.
So they say.
Okay. Thanks, Dick.
Welcome, Vince.
Alan, what have you got?
I have another set of pretty pictures. I think a few episodes back we did, was it the Nikon imaging competition?
Yeah, we did.
Well, if one optics supplier is going to do it, another one has to as well. So I stumbled across the Olympus Bioscapes 2009. They just had this contest, and they just recently announced the 2009 winners. Apparently, they've been doing this for a few years. So you can go to the website and look at these stunning images of various organisms that have been shot.
Some of these are videos.
Yes, this one's a video.
Yeah.
Third place is a Spirogyra.
This is fantastic.
Sexual attraction. What is happening here, Dick?
I believe that they're forming a
bridge.
Complete union.
Yeah.
Look at the cytoplasm is going into the other cell.
I love it.
It is an uninterrupted mating. Yes.
Indeed it is.
These are beautiful, though.
Yeah.
Wow.
Yeah, there's some really, really striking images in there. And some of the honorable mentions are really amazing, too.
Very nice. Thank you, Alan. It's beautiful. Yeah. Dick, you compare these to, what are those things out in space?
They're called planetary nebula.
Ah.
Because if you put a series of plates of planetary nebula next to these fluorescent pictures, they look very similar.
Well, there are some similarities. Yeah, that's right. I actually did that once. I called it macro, micro. Maybe I'll put some together and shove them up on the show sometime.
You have a PowerPoint presentation.
I do.
You could narrate it. I could record it, and we could put it up as a video, you know.
Why don't we?
That's what I do with my lectures.
Sure.
Look at this one here. What is this? This is gorgeous. This
Looks
is
like
a peripheral neuron from
neuron.
Singapore. From Drosophila, actually. Great. It's beautiful.
They're gorgeous.
Oh, these are nice. People will have fun with that.
Oh, that'll keep us busy for a while.
Alan.
Alan, that's great.
Okay, and my pick is something that I read every month, but never thought to share, and it's actually quite good. It's Microbe Magazine from ASM.
Oh, yeah.
This is if you're a member, you get a paper copy, but it's available online.
Yep.
At microbemagazine.org, and it's full of great stories.
No question.
This month, Dick,
yes.
New Horizons for Deep Subsurface Microbiology.
Yeah, no, I saw it. I get it too.
They take samples from 2.5 kilometers down, and they find bacteria in them. As you told me this once.
Amazing.
Is that desert varnish on the cover?
No, not at all. It's the stuff they pull out of the deep earth.
That's right.
Bits of
really?
rock. Yeah.
Because it looks striking. If you go to the Southwest and you go, like Zion National Park is famous for this.
Yeah,
these beautiful sandstone formations that have this, this discoloration on the surface, and it turns out it's microbial weathering.
This is actually fractured basalt rock in the Yakima River Canyon, Washington. Microbes living deep below the Earth's surface in rock formations and other environments are drawing the attention of researchers.
Neat.
Right.
So anyway, check it out. It's really nice. They have great articles, nicely written. A bunch of them are by researchers. They also have commentary.
Right.
This month, they also have an interesting article about symbiosis between a virus and a mycoplasma, which we may talk about sometime on TWiV. So that's my pick of the week.
Viruses are everywhere.
And that will do it for another TWiV. If you are a new listener, please think about subscribing. You can either do that in iTunes, and we're also on the Zune Marketplace.
Are we?
Zune is a portable audio device made by Microsoft, a competitor of the iPod. And TWiV is on Zunes. So you can I have a link in the show notes at twiv.tv where you can click and automatically subscribe either in your iPod or Zune. Of course, it's free, and it's great because you automatically get every new episode as we upload them. If you don't have either player or don't want to use a player, you can always go to twiv.tv. You can listen to our episodes there or download them. And, of course, we have show notes there as well, which gives you all the links to the information that we talk about. TWiV is part of microbeworld.org, a community created by the American Society for Microbiology to help disseminate information about microbes. You can also find us at sciencepodcasters.org and promednetwork.com, websites where you can find high-quality science podcasts from a wide variety of fields. I just did a podcast last week with Mark Pelletier of Futures in Biotech, which is also at sciencepodcasters.org. A lot of cool science we talked about, so check that out. As always, send us your questions and comments, twiv@twiv.tv, or you can call us at Skype and leave a message at TWiV Podcast. Now, here's a request for all of our listeners. I'd like to do an episode on viruses in the movies. So what we'll do is we'll take a movie or two and where there's a virus figuring in a prominent role. infecting people and making zombies out of them or something.
Like Outbreak with
And
Dustin
we'll critique it.
Yeah.
Okay, so send us your suggestions. I have a couple, we all do, but I'd like to hear what our listeners would like to know. What's real and what's not. Well, none of it's real, of course. But,
right.
But we'll do that sometime next year. Dick, thanks for joining us.
Vince, pleasure as usual.
I know you taught all morning, so I appreciate it.
Yeah.
But you're not done talking, right?
I am now.
Couldn't you teach another 3-hour class if you had?
I need a nap or something. Perhaps a cookie and a glass of milk.
Dick can be found when he's not right in front of me or in his office at medicalecology.org and Racaniello.org. And we'll see you next week, right, Dick?
Well,
Alan, thanks for coming back.
Yeah, it was a good time.
Or I should say, glad you're back again.
Indeed. Yeah.
And Alan is at dovedocs.com. Did I get that right?
Yes, you did.
Even though I haven't said it for 2 weeks. Will we see you next week?
I plan to be here.
Great. Rich Condit will join us next week, the 18th, for another episode of TWiV. You have been listening to This Week in Virology, all about viruses. Thanks for joining us, everyone. We'll be back next week. Another TWiV is viral.